Assalamualaikum everyone. Hope you are all doing well. I have a presentation of Principle of Chemistry 1. This is me Ariba Islam. My presentation topic is hyaluronic acids, which is submitted to Dr. Hammad Majeed. Here are table of contents. First one is introduction of hyaluronic acid. Hyaluronic acid. History of hyaluronic acid. Biosynthetic pathway of hyaluronic acid. Structure of hyaluronic acid. Biological functions of hyaluronic acid. Uses of hyaluronic acid. Potential side effects and risk. Features of hyaluronic acid. References. First, now I will you about his history. What is hyaluronic acid? Hyaluronic acid, also known as hyalurones, is naturally occurring anionic non-sulfated glycosaminoglycan found in its extracellular matrix of connective epithelial and neutral tissues. It is unique among glycosaminoglycans is that it is non-sulfated form. It is plasma membrane and plasma membrane and can be very large when its molecular weight often reaching the millions. Hyaluronic acids play a crucial role in the extracellular matrix, contributing to cell proliferations and migrations. It is a major component of skin where it is involved in tissue repair. When skin is exposed to excessive UV rays, cells in the dermis stop producing as much hyaluronic acids and increases degradations. In joints, hyaluronic acid is a major component of synovial fluid where it acts as a lubricant and shock absorber. It is also as important component of articular cartilage where it forms large aggregates when proteoglycans that imbibe water and provide resilience to compression. The molecular weight of hyaluronic acid in cartilage decreases with age, but the total amount increases. Hyaluronic acid has been used medically to treat osteoarthritis for the knee via intra-articular injection. Of 2020 meta-analysis found that high molecular weight hyaluronic acid improved both pain and functions in people with knee osteoarthritis. Hyaluronic acid is also used to treat dry eye, and a dermal filler is cosmetic surgery. It is produced commercially by extractions from animal tissue such as roaster combs, and by bacterial fermentation. It is considered safe for use in various forms like supplemented skin and products and injections. Allergic reactions are rare. Structures of hyaluronic acid monomers. Hyaluronic acid consists of glycuronic acid and an acetyl glycosamine that can be repeated up to 10,000 times or more. History. Hyaluronic acid was first discovered in 1880 by French scientist Pierre Portes, who observed that the mucin in the eye's vitreous body was different from other mucoids. However, it will not until 1934 that German biochemist called Meyer and his colleagues John Palmer isolated and identified hyaluronic acid as a purple polysaccharide containing as a minor sugar and a uronic acid. In the 1940s and 1950s, the psychochemical properties of hyaluronic acid were studied in details. In chemical structure was thought in 1954 by Meyer and Westman, who determined it was composed of repeating disaccharide units of D-glyconic acid and an acetyl by D-glycosamine. During the 1960s, scientists figured out how to extract hyaluronic acid from animal tissues, particularly human umbilicals, cords, and rostral comb. However, this approach had issues of purifying unwanted contaminants, leading to first studies on producing hyaluronic acid via bacterial fermentation and chemical synthesis. In 1979, Hungarian inventor under Habalas developed an efficient method for extracting and purifying pharmaceutical-grade hyaluronic acid from roaster comb and human umbilical cords. This marked the beginning of industrial production of hyaluronic acid from medical applications. The 1980s and 1990s saw the introduction of the first hyaluronic-based medical product, Halon, used in cataract surgery. Since the understanding of hyaluronic biological roles has led to increasing use in the variety of medical and cosmetic applications. Biosynthetic pathway. Most studies of hyaluronic biosynthetic in prokaryotes have focused on zooepidemicus, which uses two distinct pathway to synthesis hyaluronic precursors. Biosynthesis of hyaluronic begins with the phosphorylation of glucose by hexokinase to produce the main precursor glucose 6-phosphate. From here, hyaluronic acid synthesis pathway can then to divide it into two synthetic pathways that synthesize the two building blocks of hyaluronic glycuronic acid and an acetyl glycosamine. Hyaluronic acid biosynthetic pathway is zooepidemics. Glucose is first converted by hexokinase to form glucose 6-phosphate, which then enters off the two distinct pathways from UDP, glycuronic acid, PGM has C and has B. UDP, acetyl glycosamine has E, GLAMS as GLAM M and has D. These precursors are then bound together via the actions of hyaluronic acid synthesis of has encoded by has A is S. Zooepidemicus to have hyaluronic acid is the first set of reactions of phosphoglutamate PGM converted glucose 6-phosphate to glucose 1-phosphate before a phosphate group from UTP is transferred glucose 1-phosphate by UDP glucose pyrophosphorylase to produce UDP glucose. Finally, UDP glucose is oxidized by UDP glucose dehydrogenase to yield the first hyaluronic acid precursor UDP glycuronic acid. In the second set, glucose 6-phosphate is converted to fructose 6-phosphate by phosphoglycosaminase. One converted fructose 6-phosphate is tagged with an amino group transferred from a glycotamine residue via aminotransferase to produce glycosamine 6-phosphate and then is modified by mutase to yield glycosamine 1-phosphate. This intermediate is then sequentially acetylated and phosphorylated to acetylate transferase and phosphorylase respectively to yield the second hyaluronic precursors UDP and acetyl glycosamine. Once the two precursor has synthesized hyaluronic acid, hyaluronane synthesized polymerase, the two components is an alternate manner to produce the hyaluronic polymers. It is important to note that hyaluronic biosynthesis is an energy-consuming process for bacteria as several intermediates are used in cell walls, biosynthesis, biomass formations, and lactate formation via glycolysis. Now I will explain you about hyaluronic synthesis. In general, for any bacterium to synthesize hyaluronic capsule, the hyaluronic synthesized genes must be present as it needed to polymerase UDP sugar precursors into hyaluronic acid. The hyaluronic acid synthesized has first isolated from S pyogenes from group A Streptococcus, and it was shown to have the capability to direct hyaluronic acid capsule biosynthesis in the capsular mutant as well as heterologous bacteria such as E. coli and Enterococcus faecalis. Their other hyaluronic acid related genes have been identified in other organisms including group C Streptococcus, algae, bifiduses, and vertebrates. Analysis of the primary sequence and predicted structural topologies have shown hyaluronic acid synthesis enzymes share many common features and have been divided into two categories based on similarities and difference in amino acid modifiers topology and mode of action designated as class 1 and class 2 hyaluronic synthesis. Table 1. Characteristics and features of class 1 and class 2 hyaluronic acid. Features. Class 1 and class 2 features: source organism, Streptococci, amphibian mammals used virus. Class 2 Pasteurella multipodia. Features: ability to express at soluble active protein. Class 1 no must be associated with cell membrane. Class 2 yes, amino acid residues 107 sheet. Features: number of GT2 modula, class 1 and class 2. at reducing and Streptococcus humans and mice, hyaluronic acid chain growth, at non-reducing and Xenopus laevis and Elga virus, class 2 at non-reducing and features topology of the protein, class 1 multiple associated throughout protein membrane domains, the whole class 2 two catalytic independent domains A1 and A2 attached to membrane via C-terminals region. Intrinsic polymerization processive and non-processive. Features size amino acid class 1 417 to 558, class 2 972. Features primer for initiations of hyaluronic acid synthesized, class 1 no, class 2 yes. Features involvement of other molecules protein during hyaluronic translocations, class 1 yes, involves lipid molecules membrane bilayer, class 2 yes, may involve capsular polysaccharide transport machinery. More studies needed to confirm. Now I will explain you about class 1 hyaluronic synthesis. The vast majority hyaluronic acid synthesis enzyme are class 1 and have been identified in floria like green algae virus, group 1 and C, streptococci as well as vertebrates. The key features that define can 1 class 1 hyaluronic acid synthesized included class 1 hyaluronic acid enzymes have a single glycotransferases family module, subset of common catalytic residues among glycotransferase enzymes from their sugar additions reaction and share amino acid similarities in the central region of the protein sequence. Second one, this protein group is an integral membrane enzymes with 4 to 6 predicted transmembrane domains and 1 to 2 membrane domains are found in each structure. Third one, this enzyme are thought to be associated with lipid molecules in order to synthesize and extruded the hyaluronic acid molecules out of the cells to form ether. The capsule are to be released into the extracellular space. Now I will schematic representation of hyaluronic synthesize in class 1 and class 2 hyaluronic acid synthesize. Class 1 hyaluronic acid synthesized integral membrane protein that catalyze the UDP sugar addition to growing hyaluronic chain and may be transport the hydrophilic hyaluronic acid polymer across the cell membrane of eukaryote or gram-positive bacteria. Lipid molecules, yellow circles, facilitated the hyaluronic acid activity, which allows it to direct hyaluronic translocations. B class 2 hyaluronic acid is a spherical protein that also catalyzes hyaluronic elongation. This enzyme is a hybrid of two glycotransferases that transfer and the non-reducing end of the hyaluronic chain. It was suggested that this protein may interact with other cell membrane protein, capsular polysaccharide transport protein, purple block in order to translate hyaluronic across the cell membrane of gram-negative bacteria. Blue and red dots represent GLCUA and GLCUNAC respectively. Orange triangle represents the UDP component of the sugar. 4 class 1 hyaluronic acid synthesis adds UDP sugars to the growing hyaluronic acid polymer from the reducing end of the growing hyaluronic acid chain and some at the non-reducing end for Xenopus laevis and Elga virus has. Now I will explain you about class 2 hyaluronic acid synthesis. The only organisms known to and not encode for a class 2 hyaluronic acid synthesized enzymes thus far in P. maltosida, the protein known as PM hyaluronic acid synthesis has a number of key features that distinguish it from class 1 hyaluronic acid synthesis. First one is PM hyaluronic acid synthesized is approximately twice the size of a class 1 hyaluronic acid synthesized. Second one is PM hyaluronic acid synthesized has two GT2 family modules. Third one, PM hyaluronic acid synthesizes a spherical membrane protein that does not necessarily require association with lipid bilayer and transporters of cell membrane to synthesize hyaluronic acid. Fourth one is PM hyaluronic acid synthesizing the stop independent catalytic domains that are responsible for GLC, NAC, and GLC-UA transferase activity. And fifth one is PM hyaluronic acid adds UDP sugars to the growing hyaluronic polymers from the non-reducing end. PM hyaluronic acid synthesis has been expressed and successfully synthesized hyaluronic acid in a number of bacterial species, including E. coli. Since then, these enzymes have become a focal point for hyaluronic acid production, as its truncated form PM hazy 703 does not need it to be bound to a membrane in order to synthesize hyaluronic acid. Structural properties. Hyaluronic acid is a layer heteropolysaccharide glycosaminoglycan macropolysaccharide with high molecular weight formed by regularly repeating residues of an acetyl D-glycosamine D-glycuronic acid in a hyaluronic molecule. The D-glycuronic acid is associated with amino sugar by beta-1,3 glycosidic linking, and amino sugar is connected with the D-glycuronic acid by a beta-1,4 glycoside T. Obviously, that structure of monomeric united name as the primary structure is not dependent on the molecular weight of hyaluronic acid. The extensions of polar and polar segmented in the polymer structure affords hyaluronic acid capability to chemically interact with various chemical agents. For instance, with metachromatic dyes, which find applications in clinically examinations, and Ketosan, which makes it possible to obtain a new class of material based on polyelectrolytic complexes. Hyaluronic acids form hydrogen bonds, which on one hand could poise the micromolecular solution, but on the other hand give rise to rigidity in the polymer system, which finally specify the properties of hyaluronic acid solutions. Demonstrates the potential hydrogen bond that could form both within one micromolecule and between the adjoint molecules. Note that aqueous molecules could be bridged between the two connected functional groups. Eventually, such primary substitution and hydrogen bond have to form secondary and tertiary structure. Hyaluronic acid and its salt with ammonium ions, magnesium, and alkaline metals have good solubility in water and possess a high molecular viscosity even at a low polymer concentration. Moreover, hyaluronic acid solution could organize a three-dimensional cellular structure with enormous dimension at concentration of less than 1 omega ml. Bi-construct biopolymers can organize pseudo-gels when concentration are equal to or above 1.0. However, hyaluronic acid with high molecular weight equals 5.0 MD at concentration greater than 0.1 forms entanged polymers network, but hyaluronic acid solution do not prolong mechanical integrity. Salt of hyaluronic acid with cation possessing 2 or more valency number have substantial insolubility in water. Additionally, if such ions are introduced into hyaluronic acid solution, intramolecular crossing can constitute, resulting in the development of a gel with great water content. It is known that a macromolecular hyaluronic acid solution could organize the left-oriented individual or twin spiral. Study confirmed the hyaluronic acid K and Na salt demonstrated a twin helix structure in solution. Moreover, that helix has antiparallel left-oriented strands, demonstrated the antiparallel double structure of hyaluronic acid. It is interesting to note that apart from the double helix structure, there is suprahelical organization of hyaluronic acids that could also be detected, as mentioned above. Within an increase in hyaluronic acid concentration and intramolecular polymer network could be formed in the diluted hyaluronic acid solution. The biopolymers would be separated molecules, but with the increases of polymer concentration, three-dimensional web structures and organized. This is due to the incapacity of the adjacent hyaluronic acid molecules to have several varying and separate forms. Note the free hyaluronic acid molecules could be folded into the row. The secondary hyaluronic acid structure bears a likeness to flat bands transformed into a helix and into a sheet. It is confirmed the intramolecular hydrogen bond affected the structure of hyaluronic acid in the diluted hyaluronic solutions. The macromolecules have semi-rigid coiled chain and could form helix band and even helical rings. Due to forming a rigid helix, the macromolecules of hyaluronic acid attract a great quantity of water and organize the wide domains of the tertiary polymer. The hydrodynamic size of the hyaluronic acids depend on the molecular weight. The hydrodynamic size as the final result affect the polymer concentration as polymer coils start to interpenetrate each other. Now I will explain you about table 1. Limited viscosity, number and hydrodynamic size of hyaluronic acid with various molecular weights. Now I will explain you about biological functions. Many characteristics of hyalurones and a wonderful invention of evolution. Look at hyaluronans biological role in comparison with molecular evolution of other biopolymers such as protein, nucleic acid, and polysaccharide. It is now known today that hyaluronic acid actively involved into the regulation of cell division, migration, differentiate, and tissues and organ regeneration of all stages of organism, embryonic development starting from fertilization up to the birth, the period of growth and sexual maturity and the aging periods. Most hyaluronic acid is synthesized in the embryonic period, signaling pathway to they are not considered as linear sequences of biochemical modification, but rather as network of the varying levels of complexity. This network initiated by extracellular ligands with interact with the receptors of the cell surface. Hyaluronic acid is a naturally occurring biopolymer with diverse biological functions in various tissues and body fluid. Here are the detailed biological functions of hyaluronic acids. Hyaluronic acid plays a significant role in the treatment of airways diseases due to anti-inflammatory and water-binding capacities. When administered via aerosols or tracheal installations, hyaluronic acid acts against inflammatory agent, protect against hyperactivity, delay bronchial remodeling, and modify biofilms associated with chronic inflammation caused by certain pathogens in the respiratory system. Hyaluronic acid can decrease the severity of elastase-induced emphysema in murine models, prevent bronchoconstrictions in estimates, and improve functional parameters in patients with chronic obstructive pulmonary disease. Inhaled hyaluronic acid increases the volume of airway surface liquid, leading mucus hydration, enhanced mucus transports, and reducing mucus plugging of airway. Moreover, nebulized hyaluronic acid has been shown to improve the tolerability of nebulized hypertonic saline, which is an effective treatment of managing bronchial secretions in patients with cystic fibrosis and bronchiectasis. The suggestions that hyaluronic acid enhance the effectiveness of existing treatment of airway disease and improve patient outcomes. Now we'll explain you about skin functions, hydration and elasticity. Hyaluronic acid is a power humectant that retain water in the skin, maintaining its hydration elasticity. This helps to keep the skin supply and youthful looking. Second one is repair and regeneration. Hyaluronic acid play a crucial role in skin repair and regeneration by providing a scaffold for cell migration and differentiation. It also helps to maintain the integrity of the skin extracellular matrix. Now I will explain about joints function. Lubrication and shock absorption. Hyaluronic acid is a key component of synovial fluid which lubricates and pushes joint, allowing for smooth movement and reducing friction. It also helps to absorb shock and distributed pressure, reducing the risk of joint damage. Second is joint health and maintenance. Hyaluronic acid helps to maintain joint health by regulation by balance of synovial fluid and preventing excessive wear and tear on joint surfaces. Now I will explain you about eye functions. First one is maintaining eye shape and moisture. Hyaluronic acid is preventing the vitreous humor of the eye, where it helps to maintain the shape of the eye and retain moisture. This prevents dryness and irritation of the eye. Second one is protecting the cornea. Hyaluronic acid also helps to protect the cornea by maintaining its hydration and preventing damage or external factors. Now I'll explain you about other psychological functions. First one is tissue repair and inflammation regulation. Hyaluronic acid plays a role in tissue repair by providing a scaffold for cell migration and differentiation. It is also hard to regulate inflammation by inflicting by various receptors and signaling pathways. Second one is embryogenesis and tumorigenesis. Hyaluronic acid is essential for embryogenesis and is likely involved in tumorigenesis, although the exact mechanism are not fully understood. Third one is cell signaling and migration. Hyaluronic acid interacts various receptors, including CD44 or RHEMM, to regulate cell signaling and migration. This is important to process such as wound healing and tissue repair. Now I will explain you about additional function. First one is free radical scavenging. Hyaluronic acid has antioxidant properties and can scavenge free radical, protecting tissues for oxidative damage. Second one is wound healing. Hyaluronic acid is involved in wound healing by providing a scaffold for cell migration and differentiate, as well as regulating inflammation and tissue repair. Now I'll explain you about uses of hyaluronic acid. Hyaluronic acid is versatile molecules with diverse application across various fields. Here is detailed overview of its uses in different fields. Skin and cosmetics. First one is moisturizing and hydration. Hyaluronic acid is used in skincare products to retain moisture and hydrate the skin. It can hold up to 1,000 times and its weight in water, making it an effective humectant. Second one is anti-aging and wrinkle reductions. Hyaluronic acid helps to plump up fine lines and wrinkles, reducing this appearance. It also stimulates collagen production, improving skin elasticity and firmness. Third one is wound healing. Hyaluronic acid accelerated wound healing by providing a scaffold for cell migration and differentiate. It also helps to maintain a moist environment, promoting tissue repair. Fourth one is skin regenerations. Hyaluronic acid is used in skin regeneration treatment to stimulate cell proliferations and differentiate, promoting healthy skin care. Now I will explain you about ophthalmology. First one is eye lubrications. Hyaluronic acid is used in eye drop to lubricate and protect the eye. It helps to maintain integrity to the cornea and prevent dryness and irritation. Second one is surgical application. Hyaluronic acid is used during cataract surgery to reduce the risk of complications and promote healing. Fourth one is joint lubrication. Hyaluronic acid is used in joint injections to lubricate and cushions joint, reducing frictions and being associated with other osteoarthritis. Fourth one is osteoarthritis treatment. Hyaluronic acid is used to treat osteoarthritis by restoring the lubricating properties of synovial fluid and reducing joint inflammations. And that is other applications. Dermal fillers. Hyaluronic acid is used in dermal fillers to plump up flaps, fill wrinkles, and shape facial features. Second one, in surgical applications, hyaluronic acid is used in various surgical procedures to promote healing and reducing scarring. Third one is pharmaceuticals. Hyaluronic acid is used as carrier molecules for targeted delivery of medication, enhancing their efficiency and reducing side effects. Now I will explain you about researching and development. And first one is embryogenesis and tumorigenesis. Hyaluronic acid is studied for its role in embryogenesis and tumorigenesis with potential applications in regenerative medicine and cancer research. Second one is cell signaling and migrations. Hyaluronic acid is researched for its interactions with cell receptor, influencing cell signals and migration, which has implications for wound healing and tissue repair. Now I will explain you about safety and precautions. Allergic reactions. Hyaluronic acid is generally considered safe, but allergic reactions can occur. It can be recommended to perform tests before using hyaluronic acid. Second one is pregnancy and breastfeeding. Hyaluronic acid is considered safe during pregnancy and breastfeeding, but it can recommended to consult a healthcare provider before using hyaluronic acid products. The third one is radiation therapy. Hyaluronic acid should not be used on the skin during radiation therapy, and it may increases the risk of skin complications. Hyaluronic acid has a wide range of application across various fields, including skincare, ophthalmology, and rheumatology, dermatology, and versatility and safety make it a valuable molecules for various medical and cosmetic uses. Now I will explain you about potential side effect and risk. Here are 3 points which I will explain. First one is allergic reaction. Second one is infection risk with injectable form. Third one is side effect of overuse and misuse. Allergic reaction. Allergic reaction to hyaluronic acid are rare but can occur. The most common adverse effect associated with hyaluronic filler are pain, bruising, redness, itching, and swelling. These side effects are usually self-limited and resolve on their own. However, some allergical reactions including anaphylaxis and edema have been reported. It is crucial to be aware to any history of sub-allergic reaction or hypersensitivity of hyaluronic before using it. And second one is infection risk with injectable form. Injectable form of hyaluronic acid carry a risk of infection. Articular injections of hyaluronic acid are generally considered safe, but there are small risk of infection, especially if proper aseptic technique is not followed. Infections are extremely rare in clinical practice, but they can occur. It is essential to follow strict aseptic technique during administration and to aspirate and joint effusion if present. Third one is side effect of overuse and misuse. Overuse and misuse of hyaluronic acid can lead to side effects. For example, excessive use of hyaluronic dermal fillers and can cause skin irritation, inflammation, or even skin necrosis. Similarly, using hyaluronic acid product without proper guidance or without using the recommended dosage and can lead to adverse effect. It is essential to consult with a healthcare and professional before using hyaluronic product, especially if you have a history of allergies or other medical conditions. Now I will explain you about additional precaution. Pregnancy and breastfeeding. The safety of hyaluronic acid during pregnancy and breastfeeding is not well established. It is recommended to avoid using hyaluronic product during these times. Radiation. Second one is radiation therapy. Applying hyaluronic acid to the skin radiations therapy can increase the risk of skin complications. It is recommended to avoid using hyaluronic product during radiation therapy. Third one is hardening of skin and connective tissue. Hyaluronic acid can worsen skin ulcer in people with scleroderma. It is recommended to avoiding using hyaluronic acid product on the skin if you have scleroderma. While hyaluronic acid is generally considered safe, if it's essential to be aware of potential side effects and risks, especially when using injectable form or dermal fillers. Proper use, dosage, and precaution and crucial to minimize adverse effects. Now I will explain you about future of hyaluronic acid. Hyaluronic acid have garnered significant attention in recent years due to its versatile applications and promising future prospect. This review delves into the evolving landscape of hyaluronic acid, exploring its potential advancement and implications across various industries. First one is market growth and technological advancement. The global hyaluronic acid market is poised for substantial growth with projections indicating a significant increase in its market value of 2025. Technology advancement in hyaluronic acid productions process and the development of novel formulations and expected to be key driver or market expansion. The rising demand of minimal invasive procedure and faster result in it also anticipated to fuel industry growth. Second one is medical and cosmetic applications. Hyaluronic acid roles in advancement therapies in promising with applications in tissue energy, dermal fillers, and osteoarthritis treatment showing significant potential in its ability to maintain skin hydration, improve collagen production, and promote wound healing positions. It is an valuable component in skincare and cosmetic products. The development of hyaluronic paste supplemented and injectables of orthopedic disorder features underscores its growing importance in medical treatment. Third one is research and development. Ongoing research on hyaluronic acids, signaling properties, cell behavior regulations, and tissue repair mechanism continues to unveil new insight into its biological function. The explorations of hyaluronic receptors like CD44 and RHAMM opens avenues for understanding its role, its various psychological process from embryonic development to wound healing and regeneration. Fourth one is anti-aging and skincare. Hyaluronic acid ability to maintain skin moisture, stimulate elastin and synthesize and promote skin flexibility positions it as key player. Its anti-aging skincare. The renewal of hyaluronic in the skin is crucial for sustaining optimal hydration and nutrient supply, essential for delaying sign of aging and maintaining skin elasticity. Fifth one is business opportunities and market dynamics. The increasing demand for hyaluronic acid product driven by the aging population and consumer preferences for aesthetic procedure present lucrative opportunities for business in the skincare and medical industries. The development of innovative hyaluronic-based product and procedure coupled with strategic mergers and acquisitions is expected to drive market growth and create new avenues for business expansion. In conclusion, the future of hyaluronic acid is bright, with expanding marketing opportunities, technological advancement, and a growing understanding for its biological functions, paving the way of innovative applications in skincare, medical treatment, and research. As hyaluronic acid continue to play a pivotal role in anti-aging solutions, tissue engineering, and regenerative medicine, its potential to transformative impact across diverse fields remain promising. Now I will explain you about references. Now that's all from my side. Thank you.